So I want to move now to our discussion of individual diseases that especially have seen early and often dramatic responses in the context of clinical trials so far. So the first topic that I'd like us to address is acute lymphoblastic leukemia. And Michael, as the pediatrician in the group, obviously this is a disease where we've seen absolutely remarkable advances in the chemotherapy setting over decades, and certainly this is where we started to really make advances in combination chemotherapy in hematologic malignancies in general. And now we've seen these remarkable breakthroughs with CAR T cells. So can you talk about the unmet need for patients, especially with relapsed and refractory ALL?
Yes, Chris. It's very important to point out that most patients with ALL on the pediatric side don't need CARs. They are effectively treated with standard chemotherapy with cure rates in the 80 to 90% range. But of course, those who are refractory initially, those who have early relapse, those who have certain genetic abnormalities and relapse multiple times, we used to, of course, just at some certain point with patients who would relapse after transplant or patients who have relapsed multiple times, have not much to offer, and they have about a 10% survival rate. But with treating these same patients that we would have put on hospice previously, we now have 75% of them alive at 1 year and 60+% of them cured, we think, over long term. So the key thing to keep in mind is patients who have refractory disease, patients who have relapsed more than once, patients who relapse after transplant, patients who have primary refractory disease, those are the ones that we're targeting with great benefit.
So I want to move from pediatric ALL to adult ALL, and David, you certainly have a lot of experience with treating.
Yeah, we have a lot of experience
ALL.
with a product, 41BB CAR in adult ALL, and again showed a very high remission rate in the 90+%. The challenges in the adult population have been seen though, and this has been seen with increased toxicity, and I'm not sure whether the toxicity is actually worse or whether older adults are just not as able to tolerate the types of toxicity that the pediatric population can. Now this is an awkward situation for everybody because the FDA has approved this product, the tisagenlecleucel, up to age 25. So I cannot think of any difference between a 25-year-old and a 26-year-old, but yet right now a 26-year-old could not get this product on label, and I think that's really too bad. But we desperately need new clinical trials in the adult population to keep going forward because there's no doubt that this is an active treatment. I have I think a 72-year-old that I treated 4 years ago now, almost 4 years ago, that's in remission for ALL with this. And so we need to get this into the adult population.
The KITE product's being studied, as you know, in adult ALL, and at this meeting, there are data being presented which I think are really encouraging. Follow-up is not as mature as the pediatric data, but I think this will be something that will be used in adult ALL, David.
Yeah, I think people are recognizing that there's several factors that impact the toxicity and the effect. And as Mike mentioned earlier, you need to probably have some target around or some antigen around to get the T cells kick-started, right? They have to encounter antigen to grow. This can be normal B cells or it can actually be leukemia. So in our studies, we actually have more trouble in MRD only positive patients getting the T cells going, whereas if someone has a lot, more than 10%, the T cells will go. But there's also likely a correlation of toxicity in those patients with very, very high tumor burdens. So there's probably a sweet spot that you'd like people to have their disease, somewhat controlled.
An ideal tumor burden, so to speak.
And I think the ideal cell dose is yet to be determined. Now I know in the tisagenlecleucel there was no dose relationships between a relatively wide range of dose. But in our studies in Seattle, and I believe in the KITE study that will be presented, they're decreasing the dose because of excessive toxicity at higher cell doses. So I think we still have a lot to learn about, what's the right T cell to use, what's the right dose of CAR T cells to use as we're treating these patients.
Yeah, and there will be differences among the products.
Yeah, products are different.